Hello, heroes. Today we're going to be doing some bioinformatics, some molecular modeling. And so if you haven't done molecular modeling yet, I strongly suggest you go and watch the part one and part two introductory molecular modeling videos on the Amino Labs YouTube channel. You'll definitely want to get up to speed on using the software because today we're just going to jump right in. Okay. So the topic of today is restriction digests, and you're going to be or in the process of doing the Digest It kit by Amino Labs. And so today we're going to do a little bit of a dive into what a restriction enzyme is. We're going to look at it through molecular modeling software, and hopefully that'll just give you a kind of a closer view so you can imagine in your mind what's going on in this process as you go through the real life bench or lab experiment. Okay. So as you remember from the other intro molecular modeling videos, Protein Data Bank or PDB is the gold standard for storing and finding molecular models of proteins and other macromolecules. And so we're going to click on that. And I also, as this is loading here, I want to remind you that you probably want to have a 3-button mouse. You can get away with a 2-button mouse on a laptop, for example, but if you have a 3-button mouse, try and get that out. Okay, so if you look at or have read the instructions for the Digest It kit, you'll know that one of the restriction enzymes is EcoR1, which is actually typed with an I instead of a 1. So if I were to go a 1 like this, it gives me like a quite different EcoR124I. So it's kind of funny with restriction enzymes, the 1 is like an I or the 3 is like 3 I's. I guess it's more Roman numerals in that regard. Okay, so if I type in EcoR1 or EcoRI, I get molecule EcoR2. I get, which is here, I get one that says EcoR1 recognition site and it's DNA. So that's not the enzyme itself, that's the DNA piece that the restriction enzyme would bind to. So this is a mutant. Let's click on this one. Okay, so this is a mutant of EcoR1 that has a mutation in it. So alanine number 138 is mutated to a T, which is threonine. All right, so yeah, this will be a pretty interesting one to look at. I'm going to download this one. Let me download the file. I'm going to download PDB format, GZ format, CIF format. These are generally all opened in PyMol. Okay, so I downloaded that one. Another one that will be interesting to look at as well, which is kind of a cousin, is EcoRV. All right, so we see a couple of these down here actually. So we've got the endonuclease EcoRV DNA complex. All right, so that's pretty cool. That would probably be pretty interesting to look at. Let's click on that, go down. You can see like the classic 4-letter code, 1RV5. I'm gonna download that one as well. Okay, so now that we've found a couple of files to look at, we're gonna go ahead and open them in PyMol. All right, so I've got a couple of files on my desktop, the 2 different files that we had just downloaded. And I have these set up so when I double click on them, they're going to open up PyMol automatically. So you may need to open PyMol first and then import them. And remember, you can use the 4-letter code to also do the importing. So yeah, let's start out with this one here. I'm going to skip the activation. go over. I'm going to make it a little larger here. And great. So this is the EcoR1 that has been mutated slightly, and it said it's a little bit more promiscuous, which means that the enzyme will bind a little more freely to other DNA as well, right? So usually restriction enzymes are quite specific to where they bind and ultimately cut. And so I guess this mutation, which again was an alanine at 138 to threonine, makes it bind a little less specifically. So maybe let's start out by, let's look at that mutation and see where it actually is. So I'm going to open up the sequence here. Let's slide over a little bit. Again, we want 138 and we're looking for a threonine. So 136, 137, 38. There we go. So we've got a T. All right, and let's go and zoom in. Heck, let's see the side chain here a little bit. So I've got it selected. I'm going to show the side chain as sticks. I'm also just going to go and show the main chain here as sticks as well. And okay, so wow, it's amazing. This little change here can make a big difference, hey? So this is, again, now this one here is now a threonine, and that makes it a little bit less specific in binding. Okay. While I've got that highlighted, maybe what I'm going to do is I'm going to look to see if there's a bond that's forming between the DNA and the amino acid here. One thing to keep in mind, actually, as we're looking at this is right now we're only seeing one strand, right? We're not seeing a double-stranded DNA. It's not a double helix that we're seeing right now. So yeah, keep that in mind. When we open up the other file, we're going to see a double helix. This one is a little bit out of the norm, we'll say. But now that we've got this one selected, let's just look to see if there's any bonds going on here with this. So we should be able to go, where is it? Find polar contacts. Remember, hydrogen bonds are polar contacts. Heck, we could just go any contact. Let's go like that. Fine, let's just go within the selection here. And what we see is, actually, there's not much going on right now. So maybe actually that was an important bond. When it was an alanine, there was a bond that was important. And now that it's a threonine, that bond no longer exists, which makes the protein less selective in where it binds, right? We see these little stars here. You might remember that's water, right? If we were to go over here, we see all these oxygens here. Those are the waters. If we were to go to the all here and hydrogens and add them, there we go. Now we see little waters, right? We've got the H2, two H's here, and then the O for all of these, and they look like little V's, right? We can also see the hydrogens now on the threonine that we showed a little bit more clearly. Okay, well, let's go ahead and select this DNA here. We're going to select all of the DNA, and we're going to go ahead and show a few things. Let's go ahead and show let's show the main chain as sticks, but let's also show the side chain as sticks for now. Okay, so now when we look at this, we can see these are the nitrogenous bases, right? So you might recognize that from the DNA, but now we also can see the backbone a little bit better. We can see some of the oxygens coming off of where the phosphorus atoms are, right? So we can see a little more clearly. If we now go and look again at this threonine, zoom in a little bit, there's still some distance there. So just by looking at this molecular model, it seems pretty clear to me now that in order for us to really understand how DNA binds to this enzyme and how this mutation makes it bind less specifically, we need to have double-stranded DNA there. And so perhaps if we were to search in PDB a little bit more, we might actually find a double-stranded piece of DNA that is bound to this enzyme, and then we could look at it. But by doing a quick search, I didn't see one. So yeah, what else could we look at here? We could just go ahead and look at, so clearly there must be some interactions between the DNA and the rest of the EcoR1 protein, right? So why don't we try and highlight that a little bit? If we were to select some of these, maybe we hide some of this because it's going to get a little bit crazy. All of this back here is not interacting with the DNA very clearly, right? It's way too far away. So why don't we go ahead and select some of that and let's hide those atoms. And so I'm on 3-button viewing mode. If I hold shift here, I can draw a box around certain atoms. Right now I'm clicked on residues. So it's going to select residues within this area here. I'm going to go maybe to there. So I could go ahead and hide all of these, but I'm just going to go ahead and I'm going to remove all those atoms, right? So we're just going to kind of chop up this a little bit until we can see only the areas that we're particularly interested in. And if I rotate this a little bit, this area is pretty far away as well. So I'm going to also select this area up here. I'm going to remove those atoms, getting a little bit closer. There's also a bunch out here. Select that area. Let's remove those atoms. All right. So it's all of this is a lot closer. So as we visualize, well, this piece here is clearly far away. Let's just delete that. Here we go. It's not super clean. You could spend a little more time cleaning it up, getting like a really nice uh, area where you have only the amino acids that are very close here. Not sure why it's still showing me this chain here. Maybe I didn't highlight it well. Maybe I'll just try and delete that. Move those atoms. There we go. It's a little bit better. We can see that this is a little high and far away. That's not going to be interacting at all. Let's delete that. And yeah, let's try that. Let's start with that. Okay, so we have a lot of waters in here. That makes it a little bit confusing when we add the side chains for these amino acids. So let's go remove the waters. That's just going to make it simpler. Um, in the other model, we'll have a closer look with the waters because sometimes the waters can actually be a bridge of hydrogen bonding between two molecules, right? So, but for now, we'll just go ahead and look purely at the bonds. All right, so now I've highlighted amino acids. Oh, it looks like there's a couple. Those ones are kind of far away. I'm just going to leave those ones off. And I am going to make sure I don't have other stuff selected. I don't. And now I'm going to let's show the side chains as sticks. Let's also show the main chain as sticks because sometimes interactions happen with both. Okay. And now I'm just going to select the whole thing and I'm going to look for bonds. Okay. And the way I do that is I'll go through the selection here. I'm going to find some bonds, polar contacts. Remember that polar contacts include hydrogen bonds, and I'm just going to do within the selection here. And these should come up as little yellow dashes, and in fact they do. You can see there's a lot of bonds over here. That's kind of what holds proteins together. But if we zoom in, we can actually see that the backbone of the DNA here, if we look in this area, let me zoom in even more. There we go. That's a good angle. We can see that this nucleotide here that I just clicked on, which is right here, this DA, all right? So it's adenine. I'm actually going to go ahead and change its color. Let's have a look at it as kind of an elemental view. So I just change the color here. Oh, why didn't it go? I have selected it. Let's try that again. Color by element. Why isn't it changing? Deselect. I reselect. Color. Strange. I'm not sure why it's not letting me change the color right now. But nonetheless, what we have here, orange is phosphorus, and then we've got red is oxygen, right? And so clearly this is a phosphate within the DNA backbone. And we can see that one of the oxygens, this one here, is actually bound to a couple of different amino acids through hydrogen bonding. So we can see there's a hydrogen here, that's what white is, hydrogen, and we've got a dash with the red oxygen here, and there's also one happening here. If we click here, we can see that this is lysine 113, right? So maybe this time I could go ahead and change its color. Let me try and change that. I'm just going to make it all magenta. There we go. Okay, so now we have colored this amino acid, this lysine, to be pink so that we can see it a little better. Let's do the same thing with this one. When I click on it, we can see that's arginine 145, right? I'm going to change that one also to pink or magenta. There we go. So those are interacting. If we scoot over here, we can see that there's an interaction between this oxygen here and this amino acid. So this is another lysine, or this is, I shouldn't say no, this is a lysine. We're going to change that one to magenta. There we go. What else we have? It looks like there's another one here. We've got this oxygen on the backbone, and it appears we've got this amino acid here, right? We can see that there's a dash between those. Let's turn that magenta. Okay, that's great. There's also another dash coming off of there. Where's that going to? Let's twist around a little bit. We can see that. So there's actually, this oxygen is hydrogen bonding to two different amino acids. This one straight above, right here it looks. Let's color that one magenta. There we go. Actually, that amino acid is also binding to two things here. It's binding to another part of the backbone right off of the ribose here. Okay, that's interesting. Right, and so you could go all the way down here. Here's another one. Let's click on this one here. There we go. We can see serine 87. Let's color that one magenta. There we go. So now we're starting to get a view of the different amino acids within this protein. They're actually really important in binding to DNA. All right. So you could do this all the way along to actually identify what's important in binding. And keep in mind, again, that this is single-stranded DNA. Normally this would bind to double-stranded DNA, but perhaps that threonine mutation has something to do with this. So as of now, we can see that, what is this, serine 85 maybe. We've got a lysine, or 87 and 89 maybe. I don't have the designations right in front of me. Another lysine here. So actually you can see just by doing some of this analysis that lysine amino acids seem to be pretty important in bonding between, here, I'm just going to click on this lysine here and this lysine here. I'm going to turn those blue. That's a little hard to see. Oh, I turned them all blue now. Dang it. Oh, that's too bad. Um, it was these two, I think. Oh, well, I wrecked it. I wanted to see whether they were also hydrogen bonding, right? And I think this was one of them right here. Actually, it is right there. It's hydrogen bonding. Let's go over here, see if Another lysine here that's blue. We'll just see if all the blue ones are actually bonding to DNA. So it's not twisting difficultly. Come on. Zoom in a little bit here. I need to get to this one right here. And actually we do see there is a hydrogen bond on that one. And one here too. All right, so wow, that's kind of an interesting insight. So you probably started this whole journey into restriction enzymes, not really knowing what they look like at a molecular level, to now you know a lot more. We can actually see that lysine residues are an important part of binding the DNA to the protein. So maybe something you should do is look at the amino acid lysine and see what is interesting about it, and see why it just by looking at its structure, why it does this. Right? Okay, so that was quite interesting. We're going to close that one and open up the other here. Remember, this is EcoRV, so this is not an enzyme that's within your kit, but nonetheless, it'd be interesting to look at it here. All right, so this one, we're already seeing a very familiar structure, right? We're seeing the DNA double helix, right? The last time we were just seeing a single strand. This is clearly double-stranded. And another really cool thing we're seeing here is that this is not continuous, right? This is actually two little mini helixes. And wow, that's cool. So this is, the enzyme has already cut this, right? And this is the cut site right here. That's pretty cool. So this is what it would look like after the DNA is cut by the enzyme, and I guess before the DNA dissociates. One thing to quickly mention here is as you go through your digestive kit, you'll be looking at some different plasmids, and you'll see that the plasmids are actually like 4,000 base pairs long, right? And in this case, this is maybe like 10, 15 base pairs maximum. So in reality, what's happening in the tubes, the reaction tubes that you're making, is there's a long circular piece of DNA that's like 4,000 base pairs, right? And this little enzyme is going to bind to it, and it's going to cut it just like this, right? That's pretty amazing. If we were to look at the bonds between this enzyme and the DNA, would you want to make a bet against me that there's uh, lysines involved? I know, that sounds like a cool activity. So why don't you go ahead and investigate what amino acids in EcoRV are involved in bonding with this DNA molecule. Okay. So before we sign off here, I guess there's one big takeaway here, and that's the enzyme, which is made up of amino acids, right? It folds into a three-dimensional structure. This protein, it's going to bump around the cell. It's going to do the 4Bs you might remember from the zero to hero journey. It's going to bump into the right spot. It's going to bump around until it finds the strong enough bonding, and you're going to figure out which amino acids are actually interacting and bonding with this DNA piece here, and it kind of locks like a key, right? A lock and key mechanism. This is the active site of the enzyme, which you learned about in chapter 6 of the Zero to Hero book, right? So this is the active center. This is where the catalysis happens, right? And remember, catalysis is the act of changing the activation energy and enabling a chemical reaction to happen that wouldn't normally happen, okay? So simply by folding around, and maybe this enzyme changes the shape a little bit of that DNA, it makes it possible to break the bonds, these phosphate bonds. They're called phosphoester bonds in the backbone. Okay. So I hope this video really enlightened uh, you in how these restriction enzymes work, what they look like, how large they are in relation to a DNA strand. And yeah, I hope as you go through the Digestate kit, you have a lot more appreciation for what's going on at the atomic level inside the tube.